Right? Or we can even say, let's say that we actually have, these pathways are actually converging. Let's just say they're even converging on more. Specifically, it's going to activate the primary motor cortex, which is going to the biceps muscles. Then let's say that the indirect pathway is going to a part of the primary cortex, which is serving the triceps muscles, right? Symptoms. Now, there is many different types of signs and symptoms. These are kind of like the big ones that you'd want to be. All right, Ninja Nerds, so what we're going to do in this video is we're going to talk about Parkinson's disease. But first off, just wanted to let you guys see the new shirt. And we're going to have other types of styles eventually. But if you guys are interested in any of our types of apparel that we'll be making eventually and having more different types of merchandise, just leave a comment down below in the comment section. Give us any type of advice of maybe the style t-shirt that you want, maybe if you're interested, and we'll go ahead and try to get those out soon and available to you guys. All right, so let's go ahead and dive right in first. So Parkinson's disease. What is Parkinson's disease? We're going to get into a lot of detail on it, but let's just try to get an overall just definition. So if someone asked you, what is Parkinson's disease? You would tell them that Parkinson's disease is the progressive degeneration of the dopaminergic neurons within the substantia nigra, and whenever those dopaminergic neurons are actually decreasing, it can lead to altered motor movement, and we'll define specifically what those motor movements are that are affected. So again, one more time, if someone just came up to you in the street and they said, what is Parkinson's disease? What would you say? You would say it's the progressive degeneration of the dopaminergic neurons that are present within the substantia nigra that can lead to altered motor movements. So let's go ahead and get into that. So whenever you're looking at Parkinson's disease, what would be some of the signs that you would be able to identify within Parkinson's disease? So there's a mnemonic out there to be able to remember signs and symptoms. Now there is many different types of signs and symptoms. These are kind of like the big ones that you'd want to be able to notice in an individual who has Parkinson's disease. So T, what does T stand for? T stands for the tremors, right? So they'll have tremors. And usually these are resting tremors, right? So they're going to have these resting tremors. Now, before I even do that, what kind of tremors are they? So we have this little guy here, right? One of the tremors is what's called a pill rolling tremor. And we'll explain what's actually happening, why tremors are occurring. But it looks like their hands are like shaking, right? And it looks like they're rolling pills within their fingers, right? So that's a pill rolling tremor. So that's one of the specific ones, right? So again, tremors is one symptom. What's another symptom? Another symptom is rigidity. So rigidity. And if you think about it, there's two different types of rigidity. There's actually another one. We're going to mention the two big ones. So there's two different types of rigidity that we're going to talk about. One of the rigidities we're going to apply to the upper limbs. And what it means is it's like a cogwheel rigidity. So a cogwheel rigidity. So what do I mean by that? If I'm trying to be able to, let's say I'm trying to pull someone's arm down, it'll go down and click, 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 click. So that's a cogwheel rigidity, right? All right, so this can also happen within the lower limbs. And what do they call that within the lower limbs? It's a rigidity, but it's called lead pipe rigidity. So it won't even want to move at all. All right, so that's going to be that type of rigidity that you'll see. You'll see two types of rigidity again. What is one type of rigidity? Cogwheel rigidity and lead pipe rigidity. Lead pipe rigidity is more common within the lower limbs, whereas cogwheel rigidity is more common within the upper limbs. All right, next one, A. A stands for akinesia. Kinesia. And there is another one here, they call it, say, akinesia or bradykinesia. So it means that there's a struggle being, so either there's little to no movement, or there's so we say akinesia is almost no movement, right? And then bradykinesia is the struggle to be able to initiate that movement. Okay, so there is two types, there's akinesia and there is bradykinesia. And akinesia is actually almost no motor movement, and then bradykinesia is very little or hard to be able to initiate the motor movement. okay? And then P. P stands for postural instability. So P stands for postural instability. So if you'll notice within these individuals, they're always going to have this hunched over posture. They're going to have this hunched over posture too. All right, and then one other thing. With akinesia or bradykinesia, because of the struggled motor movements, their face is actually expressionless. So they have what's called an expressionless face, or sometimes they even call it a mask face. Okay, so if you look here, what would we notice then? If we were to kind of look at this individual, we would notice a mask face, an expressionless face, right, due to akinesia or bradykinesia. What else would we notice? We'd notice the tremors, the resting tremors, right, pill rolling tremors. What else would we notice? Cogwheel rigidity, lead pipe rigidity. What else would we be able to notice? We'd also notice postural instability, hunched over posture. Okay, so that would give us some of the signs and symptoms of Parkinson's disease. Okay, now here's where we're going to start getting into a lot of depth. So Parkinson's disease, we're going to have to look at a lot of different nuclei. So in the actual central nervous system, we define nuclei. Nuclei is defined as a group of cell bodies that are present within the central nervous system. And then a ganglia is actually called a group of cell bodies that are present within the peripheral nervous system. But they call a lot of these structures we're going to talk about the basal ganglia, but really they should be called the basal nuclei because they're groups of cell bodies here, gray matter, that are present within the central nervous system. Okay, but they do refer to them as basal ganglia, but they should be referred to as basal nuclei. So what are some of those structures? So if you look here, this green one right here, this is actually called the putamen, okay? So this green one right here is actually called the putamen. So we call this one the putamen. This red one right here is actually called globus pallidus externus. So globus pallidus externus. And then this one over here, this purple one, is internus. So what would this one be? This would be internus. So this would be globus pallidus externus, and this is globus pallidus internus. This right here is your caudate nucleus. It's also an important component of the actual basal nuclei, so caudate nucleus. And then right here, this is just going to be your third ventricle, and you also have your hypothalamus around here. These are your thalamus, so your thalami. So these are actually going to be your thalamus right here. We'll put thalami. Over here are going to be your subthalamus. So we're going to have your subthalamus right here. And here's the big, most important one, and this is going to be the, over here are going to be your subthalamus, going to be the substantia nigra. Okay? So we have, again, what do we have? We got the putamen, the globus pallidus externus, the globus pallidus internus, the caudate nucleus, the thalami, subthalamus, and substantia nigra. One more thing before we start getting into what's actually happening. We actually call this whole thing here, putamen, globus pallidus externus, globus pallidus internus, this whole thing here is called the lentiform nucleus. So if you think about it, what is this whole thing right here? If I were to actually kind of just draw like a little triangle like this, and I have, again, that point right there, and that point right there, putamen, globus pallidus externus, globus pallidus internus, this whole thing is called lentiform nucleus. Okay? And one other thing, they actually call this putamen here, the putamen and the connection, okay, so we have the putamen right here, and we're going to have a connection to the actual cortex. And we're going to call that the neostriatal pathway. And then we're going to have another one, which we're going to talk about from the substantia nigra up to the putamen, and this is called the nigrostriatal pathway. And we'll get into a lot of these steps. So I just want to make sure that we have some terms down. And again, some of this anatomy. So again, one more time before we go into this. Putamen, globus pallidus externus, globus pallidus internus, caudate nucleus, thalami, this would be the third ventricle, subthalamus, and a substantia nigra, right? And again, this whole thing here is the lentiform nucleus. And then we do have the striatum, and then we'll have a corticostriatal pathway, nigrostriatal pathway, and then we even have what's called the striatal pallidal pathway. But we'll get into all that stuff. So first off, the problem with Parkinson's disease is abnormal motor movement, right? So if they have abnormal motor movement, sometimes what we need to understand is what is the actual normal activity of the central nervous system and how it coordinates these motor movements. So let's go ahead and do that first. But before we even get into how Parkinson's disease is occurring, let's get into the normal physiological activity here that is affecting this muscle over here. So let's go ahead and do that. So whenever you're thinking about performing a movement, let's say I want to be able to flex my bicep, all right? If I want to be able to flex my bicep, that thought for me to be able to flex my bicep had to originate usually within the prefrontal cortex, and then it actually could stimulate the premotor cortex. It could have even stimulated the primary motor cortex and even the primary somatosensory cortex. So a lot of this stuff is coming from the cortex, right? A lot of the gray matter from the cortex. So again, the thought that arises, the desire, so that thought to be able to move the muscle arises within the prefrontal cortex. The planned and repetitive motor movements that you have in there is going to be the premotor cortex. And then you can also have information sent to the primary motor cortex. And then the primary somatosensory cortex can also have fibers that come down. So let's assume that we're looking here at some of the actual cell bodies out here, these red neurons, these red cell bodies. These are going to be the cell bodies specifically of the motor cortex, all right? Primary motor cortex, premotor cortex, primary somatosensory cortex. All right, so now, what happens is, in order for us to be able to coordinate this movement, the cortex, before it sends it down and out to the muscle, it has to send this message down in to the putamen. All right, so it sends this actual neurons, these neurons that extend all the way from the cortex down into the putamen. So again, we called this pathway the cortical striatal pathway, right? Because we call this putamen part here, we call it striatum, right? So now, cortical striatal fibers come down from the cortex to the putamen. These neurons right here are releasing a specific type of neurotransmitter. That's usually a stimulatory neurotransmitter. This neurotransmitter is called glutamate. Okay, so glutamate is naturally and generally an excitatory neurotransmitter. So what it does is it releases that glutamate onto these neurons that are present within the putamen. Okay, so it releases this glutamate onto these receptors that are present on these actual neurons here, and when it does that, it stimulates this neuron. And this neuron extends all the way over here until it gets to this portion right here, in the globus pallidus internus. So it extends from the putamen all the way to the globus pallidus internus here. And this neuron releases a specific chemical. And this chemical is naturally almost always inhibitory. And this chemical is called gamma, so G, amino, A, B, which is butyric, and A, which is acid. We just call it GABA, okay? GABA stands for gamma amino butyric acid. So GABA actually binds onto these neurons that are present here within the globus pallidus internus. And when it does that, what did we say GABA is? GABA is an inhibitory neurotransmitter, where glutamate was a stimulatory neurotransmitter. So glutamate likes to initiate what's called EPSPs, excitatory postsynaptic potential, so cation influx. So it loves to load cations into the cell body through NMDA receptors, and it causes calcium to flush in, right? And causes action potentials. But GABA works through causing potassium ions to leave the cell or chloride ions to come into the cell. So by doing that, what happens? If chloride ions are coming into the cell, there's more negative ions coming in. It's undergoing IPSPs, or inhibitory postsynaptic potentials. So then what's going to happen? So if we'll look at it like this. Let's come over here real quick and look at the graph. Let's say here is my resting membrane potential. So every cell should have a resting membrane potential, right? A negative 70 millivolts, let's say. And then it has a threshold that it has to reach, in this case for a neuron, to generate an action potential. If we use red to denote the glutaminergic neurons, what they're doing is they're bringing the resting membrane potential up to approach threshold. That's called an EPSP, right? So it's trying to approach threshold. But then we use the blue to denote the GABA. It's actually bringing the resting membrane potential. It's actually bringing it away from threshold. So then what would that generate? It would want to generate a IPSP. So if you think about it, there's this mixture, right? So you can Then what's going to happen? So if we look at it like this, let's come over here real quick and look at the graph. Let's say here is my resting membrane potential. So every cell should have a resting membrane potential, right? A negative 70 millivolts, let's say. And then it has a threshold that it has to reach, in this case for a neuron, to generate an action potential. If we use red to denote the glutamatergic neurons, what they're doing is they're bringing the resting membrane potential up to approach threshold. That's called an EPSP, right? So it's trying to approach threshold for a neuron to generate an action potential. If we use red to denote, okay? In the thalamus, there's another group of cell bodies here. And these group of cell bodies like to extend all the way back up here to some of these cell bodies within the cortex. But first, before we do that, let's look at this. This guy was inhibited, right? So if you think about it, you can draw downward arrows to denote very little action potentials. Whereas this one, when he was stimulated, he's having a lot of action potentials, right? So a lot of action potentials because he was stimulated by glutamate. He inhibits him, so there's very little action potentials. So what will happen then? He'll release very little GABA. So it's not going to release as much GABA there. And if there's less GABA, then you're going to have less iPSPs. And if you have less inhibitory postsynaptic potentials, it allows for this cell body to start slowly approaching threshold. So in other words, they say it's releasing it from inhibition. So if you're releasing this neuron from inhibition, he will then technically be stimulated. And then we'll show stimulation by upward arrows. And then look what's happening now. It's going up to the cortex. And when it goes up to this cortex, let's just say it's the primary motor cortex, who will then send the information down to the muscles. This is a stimulatory effect. It's going to want to cause the muscle to contract. So this is what's called, this pathway here is called the direct pathway. Okay? So again, what's the direct pathway? Neurons coming from the cortex, which are stimulating, right? So these are action potentials. They're coming down. They're releasing glutamate. Glutamate's stimulating the GABAergic neurons, which are present within the putamen. So this is called the cortical striatal pathway. When it stimulates these neurons, they produce action potentials that run down the axon all the way through the striatal pallidal pathway to the globus pallidus internus, where it releases a lot of GABA, which inhibits this neuron. This neuron, when it's inhibited, it produces less action potentials. If it produces less action potentials, it releases less GABA on the thalamus via the pallidothalamic fibers. If this is released from inhibition or has less iPSPs, it undergoes more action potentials and stimulates the cortex to come down and initiate the movement. And again, that's the direct pathway. But like everything else, there always has to be extra. Okay. Another set of neurons can come down here. Same thing. They can come down here and they can release glutamate again. So they're going to release glutamate. That glutamate binds onto, again, GABAergic neurons, which are present within the putamen. But here's where it gets weird, right? Here's where it's different. Instead of them going all the way to the globus pallidus internus, they only go to the globus pallidus externus. And then there's actually going to be neurons here. Before we do that, what happens? Go back to this thing. This is releasing a lot of glutamate because there's a lot of action potentials coming down this. So a lot of action potentials are coming down this glutamatergic neurons. And they're releasing a lot of glutamate. Well, glutamate is stimulatory. It causes EPSPs, excitatory postsynaptic potentials. That stimulates this neuron. If it stimulates this neuron, he's going to go and actually be activated, and he's going to produce action potentials. If he produces a lot of action potentials, he's going to release a lot of GABA, gamma-aminobutyric acid. And if you produce a lot of GABA, that inhibits this neuron because it likes to cause iPSPs. So now this neuron is inhibited. Here's where it gets different. These neurons extend all the way from the globus pallidus externus to the subthalamus. Okay? So they go to the subthalamus right here. Then, again, what happened? This GABA inhibited this neuron. And if it inhibited this neuron, it produces less action potentials down this axon. If it produces less action potentials, it releases less gamma aminobutyric acid. And if there's less gamma aminobutyric acid, remember, just like this one, it's released from inhibition. There's less iPSPs and more EPSPs that are coming in. And then what happens? This neuron extends and goes all the way up over here and back to the globus pallidus internus. This is crazy, right? So if you think about it, again, what happens? Glutaminergic fibers stimulates the GABAergic fibers. This goes, what? Striatal, pallidal pathway, then pallidal-subthalamic pathway, right? When this guy's inhibited, less action potentials here, releases this one from inhibition, and this undergoes stimulation. If this undergoes stimulation, he releases a lot of glutamate. If he releases a lot of glutamate, glutamate is a stimulatory neurotransmitter. But let's draw a different neuron here. Let's actually do it like this so we have a clear cut way here. So here's another neuron so that we don't get confused. This releases glutamate, and glutamate binds onto this neuron. When it binds onto this neuron is now stimulated. And when he's stimulated, he comes over here to the thalamus, right? So again, let's follow this one. Inhibited, releases less GABA. He's released from inhibition, and this guy is stimulated. If he's stimulated, he's going to release a lot of glutamate on this guy, which is going to stimulate him. If he is stimulated, he's going to release a lot of GABA onto the neurons present within the thalamus. And if you release a lot of GABA, what happens? A lot of GABA induces iPSPs and inhibits this neuron. And if it inhibits this neuron, then the fibers that it's going to be sending up here to these guys are going to be inhibited. Okay, so now we got a good part here going so far. So let's go ahead and review this one more time, the indirect pathway. Indirect pathway, fibers from the cortex to the putamen. So it's called the cortical striatal fibers. They release glutamate. Glutamate stimulates the GABAergic neurons, which are present within the putamen. Whenever this is stimulated, he undergoes action potentials that release GABA, a lot of GABA, onto the neurons which are present within the globus pallidus externus. And if it releases a lot of GABA, it inhibits this neuron. Okay? And if this neuron's inhibited, it releases less action potentials. If it has less action potentials, it's going to release less GABA. If there's less GABA, it releases this neuron from inhibition, right? So it's actually going to stimulate this neuron, which is going to come up and release a lot of glutamate onto the GABAergic neurons which are present within the globus pallidus internus. If there's a lot of glutamate, glutamate is stimulatory. So it stimulates the action potentials down this neuron. Then if this is stimulated, it releases a lot of GABA. And if it releases a lot of GABA, GABA is an inhibitory neurotransmitter. So that causes iPSPs and inhibits the neurons in the thalamus. If this thalamic neurons are inhibited, it undergoes less action potentials. And if there's less action potentials going to the cerebral cortex, specifically the primary motor cortex or the premotor cortex, what happens? There's going to be an inhibitory action. Why is this important? So again, that was the indirect pathway, and then we already did the direct pathway. Now let me explain the significance of this, and we'll do one other thing with the substantia nigra. So if you think about it, in order for me to be able to, we were talking about the biceps, in order for me to be able to flex the biceps, these biceps muscles have to contract. But at the same time, what has to happen here? The triceps muscles have to relax. So this has to be stimulated, and these have to be inhibited. That's the purpose of this direct and indirect pathway, is that while we're stimulating muscles to contract, we have to have these muscles relax. Because if not, if there's alternating contraction or relaxation, what can that produce? That can produce tremors. And we'll talk about how that's produced. And then if there's also this contraction, this constant stiffness of contraction that gives you the rigidity, right? The cogwheel or the lead pipe rigidity. Now, we'll go into some more details on how that actually happens. But just remember, we have to have stimulatory pathways, but we also have to have inhibitory pathways. Now, how does the substantia nigra contribute to this? Now we get into it. So let's come down here. Substantia nigra is actually present within the midbrain, but for diagrammatic sake, we put it right here. Okay? But you do know that the substantia nigra is present within the midbrain of the brainstem. Okay? So in here in the substantia nigra, you have these neurons, these dopaminergic neurons. These dopaminergic neurons actually kind of come all the way out here, and they synapse on that guy right there, and the actual putamen right here, right? And then it also can come and synapse on these neurons there. Okay, so it synapses on these two neurons here. And here's what's good. Let's get in this. This neuron right here has specific types of dopamine receptors. These dopamine receptors are actually called D1 receptors. These receptors right here in the indirect pathway are D2 receptors. What's the difference? D1 receptor activates a G stimulatory pathway. So whenever dopamine is released at this pathway, what happens? He is going to stimulate this neuron. But D2 works through a G inhibitory pathway. And if you work through a G inhibitory pathway, what's that going to do then? That's going to inhibit this neuron. So now that we know that, now that we know the normal activity of this dopaminergic effect, right? These neurons can come and extend up here. If they release their dopamine on the D2 receptors here within the indirect pathway, it's going to inhibit it because of the second messenger system. And then if the dopaminergic neurons come over here and are released on the receptors of the fibers in the direct pathway, they're binding onto D1 receptors. That's activating a G stimulatory pathway, and that's stimulating that neuron. Now, if we take the normal activity, what does dopamine do to the direct pathway, and what does it do to the indirect pathway? Okay, so look. This glutamate was stimulating this neuron, and now we have dopamine coming over here and stimulating that neuron. If dopamine is stimulating this neuron, what would you expect now? The action potentials are going to be more intense. So now let's draw with the black arrow, up arrows. Look, now there's going to be even more intense release here, right? So that there's even more intense release here, you're going to be even releasing more GABA. If you're releasing more GABA, you're going to have more inhibitory input on that neuron. If there's more inhibitory input, you're going to release even less GABA. If you release even less GABA, this neuron is going to even have even more intense and more frequent action potentials. And this is going to come to the actual primary motor cortex or the premotor cortex and initiate a very powerful contraction. That's a good thing, okay? Now, let's look at what the D2 receptors do. If you inhibit this neuron, right, what was normally occurring? Well, glutamate was being released, right, which was stimulating this neuron. But now we're releasing a lot of dopamine, and dopamine is inhibiting this neuron. So if you think about it, the action potentials that are actually being produced now are what? They're less, or less than what they were before, right? So there's less action potentials here. So if there's less action potentials, are you releasing a lot of GABA? No, you're actually releasing less GABA than what you were before. If less GABA is being released, this neuron is not inhibited as much, right? So it's not going to have as much IPSPs. So it might even have a little bit more EPSPs occurring. If that's the case, this neuron is kind of released from inhibition, and he might even have a little bit more stimulatory effects. If he has more stimulatory effects, then what happens? He's going to release more GABA than normal, or than before, right? If he releases more GABA than before, this is going to inhibit this neuron. If this neuron is inhibited, it's going to have less action potentials going to the neurons within the globus pallidus internus. If it has less action potentials, it releases less glutamate. That means there's less stimulatory effect here. If there's less stimulatory effect, you have less action potentials going down this neuron. If there's less action potentials, you release less GABA. And if you release less GABA, what's going to happen to these neurons that are coming all the way up here to the cortex? They will actually be slightly stimulated. So now, if you have stimulation here, then the primary motor cortex is going to be stimulated. Why would I want to do that? Because dopaminergic effect desires to be able to increase or enhance the motor movement. That's our desire, to enhance the motor movement. So what we want to do is, so if you think about it, let's say that these neurons are coming to a part of the brain which is going to cause, specifically, it's going to activate the primary motor cortex, which is going to the biceps muscles. Then let's say that the indirect pathway is going to a part of the primary cortex, which is serving the triceps muscles, right? Or we can even say, let's say that we actually have, these pathways are actually converging. Let's just say they're even converging on multiple points here. So let's say they're converging on multiple points. You want the biceps muscles to contract really, really strong, right? So you want to have a strong contraction of the biceps muscles. So you want more stimulatory input to come in, less inhibitory input. Because this inhibitory input could also be going to the biceps muscles. So the inhibitory input could be coming to the triceps muscles, and the inhibitory input could be going to the biceps muscles. But we want as much stimulatory input possible going to the biceps muscles. That's what the purpose of this dopaminergic effect was, right? Before, the direct pathway was actually having a stimulatory effect, and the indirect pathway was having an inhibitory effect. So the biceps muscles might not have contracted as powerfully as before. Then what does dopamine do? Dopamine comes in there and says, all right, let's enhance the direct pathway, and let's even kind of kick up and increase the indirect pathway. So now what's happening to the biceps muscle? It's having more powerful contractions, right? So it's going to have better contractions here, less inhibitory input. That is a good thing. Now, one more thing, and then we're going to go into the opposite effect of dopamine. So these are cholinergic neurons. These cholinergic neurons, if dopamine is inhibiting this neuron, guess what the cholinergic neurons will be doing over here? They'll be attempting to stimulate that neuron. And then if dopamine is trying to stimulate this neuron here, guess what the cholinergic neurons are going to try to be doing right here? They're going to be trying to inhibit this neuron. So that's going to be important whenever we get into this disease process. So again, cholinergic neurons are also present. Dopamine over here loves to inhibit this neuron, right? So acetylcholine will want to try to stimulate this neuron. And that is really going to become important whenever we talk about the disease, specifically rigidity and tremors.
Hi guys, hope you are still following. This is very interesting. Thank you really part two.
Okay, so now that we've done that, we've gone over the normal physiology. Let's go over what happens in Parkinson's disease and how that affects this normal circuitry. All right? So first off, we're going to talk about some of the hypothetical theoretical causes that they believe can cause Parkinson's disease after this. We're going to talk about some of the hypothetical theoretical causes that they believe can cause Parkinson's disease after this. But let's assume that there's damage here to this dopaminergic neurons, right? So let's say for some reason there's damage. So these neurons are damaged within the actual substantia nigra. Specifically, these neurons are actually coming from a specific point. It's actually called the pars compacta. And there is another one called the pars reticularis. But most of the neurons, there's damage within the of a lot of these dopaminergic neurons, right? But if these neurons are damaged, what happens to the dopamine release here on these GABAergic neuron receptor? This was the D2 receptor, right? Because the D2 goes through the indirect, the D1 goes through the direct. D1 loves to stimulate, D2 loves to inhibit. And that normally helps to be able to enhance the movement within the direct pathway and try to increase or decrease the inhibition released onto this neuron then. Less dopamine. It's going to have less stimulatory input. If it has less stimulatory input from dopamine, then this actual action potential, we can say that the action potentials of these GABAergic neurons will drop a little bit. So we're going to say that the GABAergic neurons, their action potentials here that are coming from the putamen all the way to the globus pallidus internus is decreasing a little.